Welcome everyone. Good afternoon. Today our first speaker is Dr. Bodie Pennisi. Dr. Bodie Pennisi is a professor and a statewide extension landscape specialist in the Department of Horticulture at the University of Georgia. She received her Master's of Science in 1996 and doctoral degree in 1999 from Environmental Horticulture Department, University of Florida. She joined the horticulture department at UGA in 2000. Dr. Pennisi is responsible for planning and coordinating a statewide extension program to support the professional landscape industry and conducting applied research with emphasis on sustainable landscape practices such as biodegradable containers and microbial inoculants. Dr. Pennisi also conducts research on quantifying and qualifying floral provisioning resources for pollinators, natural enemies, and ecosystem services in urban and agricultural landscape context. Dr. Pennisi's publication record includes 25 journal articles, 58 abstracts, 75 extension publications. Dr. Pennisi, the floor is yours.
Thank you, Shamat. Can you all see my screen?
Yes, perfect.
Great. Thank you again. It's a pleasure to be here as a speaker. As Shamat mentioned, we work together and have worked for quite a few years now. I have been at UGA for 22 years. So today I'm going to talk about wildflower habitats. Wildflowers is a subject that I have been really passionate about for the last 6 years or so, and have done a fair bit of learning on my own to what those plants are and how they grow. And there is just so much that I don't know. I could spend another 20 years and I feel like I'm still scratching the surface. There's a lot of interest in wildflower habitats. And with that, I just decided that I'm going to go ahead and just put together a talk that shares some of the research that I have been doing and also just give you an opportunity to ask me questions. So with that, I'll start with saying that the wildflower plantings, and that's just a general term, I'll spend more time talking about what these are, have become quite popular. And one can say that they have always been popular because we have always tried to beautify our landscapes, our roadside, and various entities from the state, the Georgia Department of Transportation, and all sorts of, civic entities on the municipal levels have always been interested in creating aesthetically pleasing green habitat spaces. But it just seems to me like in the last 10 years, particularly with this really great awareness that has resurged about how important habitat is to supply ecosystem services, and that is to help pollinators and other beneficial insects survive and thrive. And it's certainly something that a lot of research has been going on for the last 10 years, for sure, on an international level. So what are wildflowers? I mean, wildflowers is just such a big term. And it seems to me that people don't really recognize all that wildflowers are. So for one thing, you can recognize that something that is growing on the roadside that is in bloom at any given point in time is probably going to could be called wildflower. It's going to be a herbaceous plant, meaning a soft plant that will die to the ground each year. The picture, on the left, is the habitat I established on our campus around our water pond in the research and education gardens. As you can tell, this is a coneflower, a perennial native, included in many, if not all, mixes of wildflowers. On the right-hand side is a planting of a native plant, which is not intentionally planted. This naturally exists. This is a false foxglove, and it blooms in the fall along with asters and goldenrods. And this is a plant that we have growing all over in any habitat in Georgia in October, starting probably in about beginning of October, maybe in South Georgia a little bit early, and really going to first frost almost. There are several species, and this is a wildflower still, though. Even though it was not intentionally planted by people, it is there, and we can recognize it as such. So what about weeds? I mean, we talk about wildflowers, but some of them may have a little bit of murky interpretation by some groups. So here is a publication I found, and it is by our cooperative extension service that we put together. It is titled The Weeds of Southern United States. And what was my surprise to see that the Asclepias tuberosa, which is a butterfly milkweed, it's like the poster child for bringing monarch butterflies and providing resources for pollinators. 35% of plants that are listed as weeds in the US literature are actually natives to the United States. So really, when you talk about weed, we have to recognize that this is a human-centered concept, that we as humans determine that a plant is a weed when it does not grow where you want it to grow and appear. And if you're going to expend any energy in taking that plant out of this habitat that it's growing, then most likely that's going to be a weed. Whether this happens to be a rose or it happens to be butterfly milkweed, that is entirely up to you. The plant does not have a name by itself. It's where it's growing and how you view it. So the benefits of wildflowers have really been researched, and this is very well known, that they improve soil microflora health. They attract biological control agents, which are all these natural enemies of pests. They provide forage, or like I mentioned, the butterflies when they're still caterpillar stage, and they also protect overwintering insects. Say that native plants in particular, when they're part of the wildflower, they are more beneficial to native pollinators because they have co-evolved with them. And they are more easily recognized as sources for pollen and nectar or floral resources by these pollinators and beneficial insects. In many cases, these plants, when established well, perform better than exotic plants, and they're more drought and disease tolerant. But again, this is something that you definitely have to take on a case-per-case basis, and you cannot just say that all native plants are going to be drought and disease tolerant more than exotic plants if they have not been placed in the right location or treated right. So with wildflower plantings, we have many challenges. It's very interesting how a simple bag of wildflower seeds can create such a different response depending on where it's placed. Certainly something starting from a seed mix that costs you $1.50 is going to be more desirable than something that is several $100s to fill a whole space that you have in your garden. And most people will opt for a seed starting material. Sometimes it's also what you can find. Sourcing of native plant material. But in many cases, there are certain species in those mixes, for example, the Asclepias tuberosa, which is the butterfly milkweed, and in this case here, I'm showing you anemone, can be quite expensive, almost $1,000 a pound of seeds. These wildflower plantings really don't look all that colorful and well after several years. They have a tendency to form what we call a monoculture. Just one or two plants take over the mix, even though a mix may have up to 30 species in there. So I like to say that these wildflower plantings very quickly quit looking like Monet paintings and more like something a little bit different. So I'm going to, toward the end of my presentation, if I have time, I'll share some more pictures of this really wonderful site that I have a pleasure of working at. This is at the Johns Creek in Georgia, Rivermont Golf Course. And I work with Mark Hoban, who's the superintendent there. And for quite a few years, he's been letting me play there and try different things. But he started things on his own as well. So this pollinator habitat that you're looking at there, and you can see that there's a little bit of orange here in one spot and a couple there with some blue. This was established by him. I wasn't working at the time with him. So he got seeds from a pollinator habitat, which is a mix that a company would just give land managers who are interested. And the seed is free, and then they're supposed to get it all started. He called this pollinator habitat, did it in 2014. And this picture was taken in 2018, 2019, and it's been looking like this ever since. So again, monoculture, because we're actually what we're seeing is lots and lots of goldenrod, which was not part of the original mix. So in effect, we have quite a few of these things developed, and they are challenging. In addition, I'll spend some time talking about herbicide options for these habitats, but they are limited. Germination on the sides of the species, if you have, say, 25 species, germination can be erratic. Some things may not come out of the seed mix at all. And the establishment varies from a site to site to from season to season. It is, again, biggest concern with wildflowers and the weeds, what we call weeds, again, that common that that label that we put, it's not species that we like to see growing, but nevertheless, they're coming either from a seed that is present in the seed bed or some vegetative material, some root system that is naturally there. So challenges are quite a few. I'll briefly go over some of the research that I've done in the last 2 years with a graduate student of mine who graduated last year. What we know at the time, it was 2 years ago, what we don't know is that wildflower bed preparation is definitely a critical aspect of the success of long-term habitat management. And various bed preparation methods have been studied, such as grazing by animals, solarization, which is closing the bed in some plastic and raising the temperature to about 140 degrees Fahrenheit so that you really do steam and reduce any kind of live material, both from biological standpoint, from a seed, bacteria to pathogens and any organisms, even insects that you don't want to have there. Burning also is a very valuable way of preparing the soil, herbicide applications, and also tillage. Various people have done good work to define more and more on what makes the best way to start a habitat. I'll tell you right away that nobody really knows. There hasn't been a magic bullet to all of this, a magic answer. So don't be surprised if at the end of my presentation, I don't have an answer for it. We'll just keep on finding out more and more details. There was a group of scientists in Virginia Tech that studied this a lot longer than I have, and they really dug into this. How can we establish these pollinator habitats with organic and no-till seedbed preparation techniques? So they looked at the first study, and it was done in like 2018, I think, 2017 and 18. They looked at 2 perennial grasses and 9 forbs. So that was the mix that they worked with. Perennial grasses are things very often little blue stem is included in this, and also switchgrass. And 9 forbs, again, these are the first treatment that they had in that study was that they had a no-till seedbed preparation treatment, and they just applied glyphosate just to kill anything in there, but they did not do any kind of tilling. So minimal disturbance of the seed bed so that you don't allow any of the weed seeds to emerge. Second treatment was a standard rotary power till before seeding. They found out that in that mixture of annual, biennial, and perennial wildflowers, they were more successful in a no-till with glyphosate herbicide system than in a till system. They found no correlation between the seed mass and the effects of tillage on the wildflower establishment. The same group later on decided to dig in a little bit more and take a look at some of the herbicide options and see if it makes economic sense and if it will help if you apply certain herbicides to help those wildflowers. So they actually had a little bit more mixes this time. They had a perennial annuals and biennials mix. They had one mix that was tolerant of imazapic. And finally, only a perennial mix. The first treatment was, again, no-till with glyphosate. The second treatment was repeated conventional inversion tillage to keep bringing the weed seed and then killing it, again, with total, like a glyphosate type of treatment. So like over and over, just trying to get rid of as much of that weed load that is present as possible. And then the selective herbicide imazapic was tested in this tolerant post-planting mix. And a graminicide, cefoxidim, was tested in the other 2 post-planting. They found out in the no-till plot that wildflower cover and bloom counts were greater. They concluded that herbicides were not worth the cost due to variable record of effectiveness. The effectiveness of the no-till preparation relative to the tillage contrasted with the previous research, but supported the results of their own previous study by the same research team. Well, it's worse. This was that. I found another example of a successful establishment of multiple wildflower places for research, and this was done in a really excellent article published several years ago by a group from Florida, Michigan, and California was also part of this as well. In Florida, they did mowing in 2 applications of glyphosate. After the second application, the site was burned to remove the thatch and then very shallowly tilled to break up the remaining root mat and help expose that bare earth, which is really essential to make sure that the seed is coming in contact with bare earth. Prior to the seeding in December, they raked the site lightly one more time and then rolled it back, and then put the seed with sand, broadcast it, and then rolled it again just to ensure that seed contact. In Michigan, things are a little bit different because, again, the climate is completely different there. So things started quite a bit later in the season compared to Florida. But by and large, they followed very similarly, and both were quite well established. California was another site that they established. There they did solarization. Each state decided to do the preparation of the bed a little bit different. But in all of these sites, they actually had really, really good establishment. Again, the work is usually no more than 3 years, 4 years at most. And then scientists usually don't go back to see what happens. So this is where I started looking at things like, what about plugs? So we're seeing more and more plugs in commercial nurseries, and quite a few native plants are available, native grasses and native forbs, landscape plugs ready to go in the ground. And so from there, I started thinking, well, maybe I should try to look at some other ways to do this. So we were fortunate to be funded by the CEO of Chick-fil-A, Dan Cathy, who was very interested in wildflowers and wildflower habitats. And he allowed us to work on a site that is associated with Trilith Studios. And for those of you who know anything about this, Trilith Studios is where all the Marvel pictures are filmed in Georgia. This is a wonderful location in Fayetteville. And I spent 2 years going there quite often to take data. So this is where we had, so the bottom here, this is the kind of like aerial view of the site. And this has been a farmland, rangeland for many years. But this kind of bald spot, I used to like to call it a bald spot, was actually there the entire time and had been there. So this is just a caveat that I want you to keep in the back of your mind as I'm going through the presentation. The objectives of our study was to determine how bed preparation impacted establishment, growth, and flowering of 50 native forb species. We also wanted to look at how these bed preparation will affect the weed presence and evaluate the effects of the bed preparation on arthropods that were present in these locations. So the two treatments that we did, first was tillage with Dazimid, a granular fumigant with the commercial product Bazimid. The way you do it is you till, you apply 400 pounds per acre, till again at 6-inch, water and seal with a 1-inch irrigation event. You can cover with plastic for 12 days, or you can just seal it with that irrigation event. It completely sterilizes the soil and eliminates weeds, grasses, nematodes, and any diseases. And the other treatment that we had was no-till with glyphosate. So pretty similar to what the Virginia team conducted. And here are some pictures of how the site was prepared. And I love that red soil. It was like a Martian land out there. It was a lot of fun going there and taking pictures and sending it to my colleagues. And they're like, are you on Mars? Because it's just so red there. That Georgia clay was front and center backdrop. And again, we were fortunate to have the landscaping team working there for us. And that was a great way to do the research. There we go. So this is showing you how we put in the plastic around each of the beds. After 13 days, you can see on the left side in the center picture is the no-till with the glyphosate. And after 13 days, it was till with Dazimid, and how clean the till with Dazimid site looks like. Again, we did not have the kind of weeds that you'd see in other sites, but we nevertheless did have a good presence of weeds in terms of variety of weeds. We had grasses, we had blackberry, the typical, we had some other things there too. So as soon as COVID hit, we were out there working. So we had plenty of time to be outside and do our work. And we planted the plots in September. And end of September, October, the next day was watering. Spencer Shelton is my student, and I have a picture of him later on. We planted 720 4.5-inch plugs with him, and they were set on a triangular setting about 3 feet apart. And each of these, this is for scientific purposes. So the species that we looked at were representing quite a few families. It was important to look at a variety of different kinds of plants that flower for the entire season. We selected things from March to October. Research Asteraceae, which are the daisy family, and the mint family, which is the Lamiaceae. We also had quite a few other families involved as well. They are all native to North America, grown from seed. And I grew them from seed myself, learned a lot about propagation of these plants and what they like in the greenhouse. Easy enough to grow. I didn't really have any trouble with them. You could see the scientific name and the common names, the asters. We had quite a few: coneflower, frost aster, smooth aster, vernonia, which is also tall ironweed, another aster type. And then we had some others. So again, a good combination of plants. And this is what the site looked like. So the top is a picture shows you tillage with dasomet. So these pictures were taken in the summer, so 21. After we planted in 2020 in October, they went through 3 irrigation events that it simulated. So we applied 1 inch of water 3 times. And after that, nothing else. And then, again, that had been already months after the bed had been prepared, which was in May, as I said earlier. So the top is the tillage and the dasomet, and the bottom is the no-till with the glyphosate. So let's take a look at the numbers of these plants. And so we definitely found an effect of the bed preparation, which just shows you that when you see a number that is like highlight this 0.001, which means in scientific lingo that this was highly significant, meaning that statistically we show that they really did make the treatment did make a difference. So bed preparation really affected it. The kind of species that we used was also affected, meaning that each species did not perform same as the rest. So there were differences. And it also depended on an interaction between these two, which were the kind of bed preparation and the plant species. So let's try to unravel a little bit what this means. When I mentioned plant morphometrics, I mean the growth index. So briefly, how big the plant got. And the number of blooms, straightforward. If you don't have blooms, you're not going to bring in any insects. It's a straightforward correlation there. So the tillage and the dasomet, you could see how much larger these plants got compared to the non-tillage and the glyphosate. And the same thing with the number of blooms, almost twice the number of blooms. That's across all species that were used. So 720 plants. In terms of the effect of the bed preparation with the dasomet in particular, A, C, and E show you the beds that had been prepared with the dasomet, so tillage and dasomet, very little germination of weeds at all. And the B, D, and F are showing you the germinated weeds. So we definitely did have some weeds that germinated, but they did not appear to grow much. And these are just some numbers showing you that the number of weeds, quite a bit of difference between the no-tilled and then the tillage with the dasomet. And these numbers in the bottom table just were electronically generated by me looking with Spencer of how much of that green mass in each of these quadrants belonged to a weed. So that was calculated through a computer. Clearly, a lot more weeds, both in coverage of the area and the number of weeds, which was the first table. So unpacking this a little bit more in terms of which species perform better under, in other words, combine both treatments. That's how you could look at this. So the growth index of the frost aster. So frost aster performed the best. So these are arranged according to the growth index, not according to the number of blooms from highest to lowest. And you could see that the top 4 really belonged to the aster. So they did really, really well. And then followed by rattlesnake master, which was another plant. And you can also see how their blooms performed as well. And then as we look a little bit more closely at the individual, because the combination made a difference. So statistically, we actually have to look at each of these blocked. So it's like tillage and dasomet. How did that affect each of the species? So because it indicated that in some combinations of tillage versus non-tillage, species were not affected the same way. So how were these species ranked when we only looked at within the tillage and dasomet treatment? So pretty much the same. We had the frost aster, the tall ironweed, and the false aster being on the, but the rattlesnake master kind of inched a little bit higher here when we looked under the tillage. So that means that the rattlesnake master preferred to be in the tillage and the dasomet. And the lowest one was that ditch stonecrop, which is not a plant that most people will recognize, but nevertheless, it's an interesting one. And it is quite easily included in mixes. So we looked at the no-till and the glyphosate. So that would be like the standard, like you're not going to touch the soil with anything. Frost aster still did the best. And tall ironweed became better than the false aster. So when it was tough, that is no-till and glyphosate, we didn't, push the weeds with any other way, and the compaction of the soil, you're really seeing how the tall ironweed got better growth than false aster or any of the others. So that's how we should be looking at these. The discussion that in the results of the plants were grown in beds prepared with tillage and the dasomet were larger, they produced more blooms, and they faced less weed competition compared to their counterparts that were grown in the beds that were untilled and treated with glyphosate. Dasamet was effective in suppressing the weeds more than 18 months after its initial application. The weed counts were 4 times greater, and weed coverage was more double in the no-till and glyphosate plots. Since both preparation treatments received the same amount of natural irrigation, soil compaction was most likely the factor that caused the growth reduction in that no-till and glyphosate treated plots. We did some measurements on the compaction, and it was significant in the no-till. And again, a lot of it had to do with the fact that this site had been exposed, the soil had been exposed and really compacted from the irrigation. All 15 species bloomed the first year, and their combined phenology, meaning that flowering time, spanned March to October. The top performing species in both of these treatments were, again, the frost aster, starting from the left, the tall ironweed, false aster in the middle, or toward the right, with a white bloom there, and the orange coneflower. And we all have, the 3 species, the frost aster, the vernonia, which is the ironweed, and the orange coneflower are native to Georgia. The boltonia, which is the false aster, is not native to Georgia, but grows exceptionally well. And I have planted it now in other habitats, and I have to vouch for it. It's a really great plant. As far as the arthropods were concerned, we saw quite a few arthropods, lots of wasps, lots of bees, natural enemies such as spiders, syrphid flies here, and again the bees. And there is a ambush bug that had killed a bee right here, and that's the way life goes. You have a predator and you have a prey there, and that's a healthy food web that existed there. So bed preparation did not affect the number or diversity of the arthropods, and the wildflower planting supported quite diverse community 10 insect orders, including pollinators and, again, other beneficial arthropods. So this is just to show you the top picture is of one of the plants that is probably the earliest bloomer, which is the golden Alexander, scientific name Zizia. And it blooms like in March. You start seeing the blooms. And those are some of the earliest bees that we saw on these blooms. And the bottom picture shows you a Andrina, which is one of the native bee genera. And that's a female bee. And she was digging in a site. So she brought in her home right there. And that was a great thing to see. So we had good establishment of the arthropod community as well. So this is nodding onion, which was one of the favorite plants that I have been working with. It is kind of medium, like June grower and flower. Well, June, grows and flowers quite a bit in that period of time. Lots of bumblebees and then some other bees as well. So there's a video of 2 bumblebees and a little tiny bee, which was one of the sweat bees that seemed to like it quite a bit. So nodding onion is native to Georgia, and the name nodding comes from the fact that that little fruit that you see, that hook on that inflorescence stem is pushing it downward. So that's why it's called nodding. Rattlesnake master, great plant again, tough plant. It's kind of hard to imagine that it's related to things like dill and fennel, but it's in the same family, and carrot. So it's a rattlesnake master. And this is the video of rattlesnake master bloom, and there is a spider, and there is that ambush bug right here, and there's a bee. One interesting thing was at the end of 2021, 91% of the plants in the till and Dasamet plots were alive, compared to 85% of the plants in the no-till and glyphosate plots. And considering how tough the plants and the environment was with that exposed site, it was really amazing to me that 85% of those plants survived. And even in the no-till, they may have looked smaller, but nevertheless, they were still alive. A good indication that they would survive as they continue to establish. So we might ask, well, what happened this year in May of 2022? Well, in May, things looked like they were waking up. You could still see suppression of the weeds in those plots that were treated with Dasamet. And this top picture shows you one aster and how wide it spread. So beyond that, it's almost at 24 inches, 2 feet away from the original spot where it was about maybe, 10 inches, 12 inches across. And so that tells me that this plant is actually getting ready to establish a colony really, really well. And this is the growth in May of one of the rattlesnake masters, so quite good. This is some of the butterfly milkweed poking up. And again, these plants were separated from each other, so they were not close together. 3 feet apart just to allow for this. But then I got a call and they told me that I have 2 days to dig up any material from that site because the movie studio was running out of parking space. So guess what happened? We went out there, we dug up the plants that we wanted to save, which were the rattlesnake master in the left, and then the Asclepias tuberosa, which was starting to bloom. And then I salvaged them, put them in the greenhouse, and lo and behold, every single one of them survived and turned into a beautiful, beautiful plant. So the conclusion for this research, which was that the 50 native forbs were grown in bed, prepared with tillage, and desmate were larger and produced more blooms. We determined from our research that desmate was an effective tool to suppress weed emergence. And from the 15 species that we can recommend for long season flowering would be the frost aster, the ironweed, and then the Boltonia, which performed well in both of these treatments. And they established really well, all 15 of them from plugs. And then the bed preparation did not seem to affect insect activity or the abundance. And this was Spencer Shelton, who successfully graduated and now is gainfully employed. And he was out there measuring plants and doing all that he needs to do. All right. For the remaining time, I'm going to share a little bit about some other project that I've been involved with. This one is the President Jimmy Carter's solar farm in Plains, Georgia. And for those of you who haven't been down to Plains, you got to go and you have to sample that peanut ice cream. It is just absolutely yummy. But I go there quite a bit now with this project, have been going for 3 years. This is a 7-acre wildflower habitat that was established in 2018. They started in 2017 with soil preparation. So they did quite a bit of soil prep to get the soil established. But this is a 7-acre solar farm and lots of partners. We work with industry, with Earn Seeds, and with Sol America and NRCS and USDA Fisheries and Wildlife Service and some other entities such as the Pollinator Partnership. So good partners, and we're really excited to continue working there. What we did is 2 solid years of multiple cover cropping and repeated tillage with glyphosate to keep reducing the weed lock, the weed pressure in this site. So in 2017, 2018, again, repeated red clover, annual ryegrass, buckwheat, and then the wildflower seeds were placed in October of 2019. And what we wanted to see there is which one of these 2 mixes is going to perform better compared to each other, and then the control, which was just whatever germinated from the spontaneous weeds from what was left there. But the first would be the economic low diversity pollinator mix with 7 species. And then we had the high diversity pollinator mix with 18 species. And again, all of these were all native to Georgia. Full sun plants with 1 grass, which is the big panic grass, and everything else in the mixes were forbs. This is April 2021 on the left, and you could see how well the red clover has established. In May 21, so once that really healthy clover is senescing and setting seed, then we're going to get to see some more of the wildflowers starting up. So this is Coreopsis here. As we go into June, we saw some Asclepias, and you could see low diversity mix and high diversity mix. A lot of what you're seeing here, the green and the high diversity mix, this is coming from the yarrow. In my opinion, we had a lot of weed pressure. Even though they had done 2 years of cover cropping, the weed pressure is quite significant, and we're spending quite a bit of energy on treating with graminicides, graminicide herbicides, and also imazapic, which I had mentioned earlier for broadleaves, and also some woody plants and things of that nature. But a little close up on the southern beer tongue, which is one that started blooming last year very well. It's a perennial plant. It's somewhat of a June bloomer, while the rest of them are more, other than the yarrow, which blooms about the same time. In the high diversity makes the yarrow, which is that ferny looking foliage. Morning glory was significant there. And we have also cedar, we have ragweed, and we also have a lot of verbena, which is that Brazilian type of verbena from South America. So it's not a native plant, but it's very much there. And there are some pictures here. The picture on the lower left is showing you the southern beer tongue and the low diversity mix. This is the picture of the pink that you saw earlier. Those are plants and how big they have established, which they were doing a good job. The other species that established well was a southern elephant's foot, which you could see here. Again, they're getting just larger and larger clumping. They're not really much to look at as far as flower. The elephant foot is flowers at about 24, 26 inches in height. And it's a smaller kind of flower, but it is a tough plant. So those are some pictures from that. In October last year, when we went, we saw lots and lots of that blue mist flower that was inside the array. And this picture with me is outside of the array, but there was no mowing. What we had to do last year was implement more rigorous mowing schedule, which really knocked a lot of the blooms. And I think it impacted our bloom presentation. But definitely we had quite a few plants established well. Here is the blue mist and the yarrow, what I consider the bulkier type of plants and the high diversity mix. In my opinion, they successfully pushed against any of the weeds, and they would probably in the years in future would do a better job competing against the weeds. This is from this year. Lots of flowers of the clover. You see a lot more yarrow here, lots of white blooms. And on the outside of the array, just to show you that we didn't see any of the Rudbeckias or the Gaillardia, which are the Indian blanket, and the yellow here inside the array. We don't know why, but we just did not see what we saw in 2020 and 2021 even. So these two species, even though I do see them germinating on the ground, perhaps even some of the herbicides that we're applying may be knocking some of those plants down. We don't control anything on the outside. This is just all the mixed seed outside of the array, but in the array, because again, we have to consider things not interfering with the solar energy, with the panels. So there's a lot of chemical control that is done underneath the panels, around the power boxes and the inverters, to full 100% suppression of any vegetation, and then also mowing too. This solar farm is in the urban matrix, so there are houses right behind it. And last year, we had a conversation with the fire chief right after the clover leaf kind of went senescent and it turned into a big standing hay looking thing. So the fire chief was concerned that we're going to get some problems. So we had to mow, and I think that impacted our development of the plants. So again, this is from this year. Lots and lots of that Achillea, which is the yarrow, really, really well established. And some of that, again, it's almost like it survived here underneath the panels somewhat, but nothing like what we see outside the panels. What I learned there is intensive management is necessary, but it's going to affect what kind of plants you're going to see. And here is again the southern elephant foot this year in bloom. You can see this is how tall it gets. It's almost getting to a point that it's going to be underneath the panels. There is some pigweed, which we have plenty, and look at how big some of the ragweed is getting. So again, we have a lot of weed pressure, even after 2 years of cover cropping and repeated glyphosate applications before the seed was put in. Here I am holding one of the baptisias, which is a wild indigo, and there were just a few in the whole side that I could find. And the rest of what the green mass all around me is the yarrow, which is a really, really well-established plant. And this was from October 22 this year. Asters, naturally asters that were present there. And we mowed outside. They were, again, these plants were not planted by us. They're native. They just come out, and they were buzzing. The whole field was just absolutely covered with those asters, and lots of bee activity, wasps, and all sorts of butterflies and flies and things like that. But again, it was outside. And this was mowed the same time. So it just goes to show you that if it was mowed in end of August, you still can get really, really good presentation or bloom. Just a few little photos here. Just this is a aster, which is similar to what I was just showing in the previous slide. And this is the elephant foot right there next to it. That yellow could be a phytotoxicity. There is some more here in the top picture, but it could be also senescence, and it could be also drought response too, because it was getting kind of dry. And then that's the arrow, again, spreading really well. As far as how they did, the smaller green arrow means that they did really good. And if it's a smaller green one or larger one, it's lots of them, and green is smaller or fewer plants. Again, this is the mist flower established well, the Indian blanket flower the first year. So this is aggregate from 3 years, and the common yarrow established really, really well. And so did the southern elephant foot. But by and large, most of the plants did not really establish in the numbers that we felt like we would see. So what I learned there is even intensive management of a site is not a guarantee that all the species seed that come in the wheat, the seed mixes are going to come out, and B, that they're going to really, establish well. And I don't know if I have time to go through this, probably not, but just a quick picture again. Mark Hoban, that's superintendent in the Rivermont Golf Course. We did a quick work there in March of 2020. We started this site with some wildflower seed mix, and they were just placed so that we could evaluate them with working with some soil treatments with biochar and mycorrhizae. And again, this is me in June of that year, just forlornly looking down on the ground at basically nothing but weeds. So we did not get anything to just about none of the seeds established or grew, but we think that it was because of the excessive rains that we got from the hillside, and it just washed. And there's a creek here to the left. So I think that had a lot to do with it. And so this year I put in transplants. I just went ahead and I transplanted about 100 or so transplants. I just said, enough. We're just going to put some transplants in that spot. And this is Rebecca, who is helping me battle the weeds there, which is the dog fennel was really, really huge pressure. But in between there, we went there a couple of times and we weed whacked around the natives. First, we had to find them. And here is me pointing out that this is actually one of the planted mountain mints that I put in here. So it was easily about 30 inches. And with all the weeds around it, it just pushes the plants to grow taller. So there's the cluster mountain mint after we cleaned all around, weed whacked around it. So there is also the shrubby St. John's wort. Really, really good establishment of that plant. Don't pay attention to this. This is a native plant, but it is not one that we planted. Sawtooth sunflower. Nodding onion, again, this is that patch right here. Hairy sunflower, sweet Indian plantain, and calamint. So orange tags, you have to put the flags in order to see where the plants are. But by and large, all the plants I put down, they were irrigated only one time. I don't think that we irrigated them anymore. Again, this site, we decided to do something similar. We decided to clean and as much as possible preserve the desirables, that is a blue indigo. Many of those plants were right in this area. Lots of pressure from goldenrod, from even woody plants, poison ivy all over the place, blackberry. I mean, really, really strong pressure. And here is me after we managed to have enough space for me to squat down next to all the poison ivy. But this is the blue baptisia again, quite a bit of pleasure to see. So with that, I've finished, and I'll invite any questions. And thank you for your attention.
Thank you so much, Bodie. This is the time for any questions that you might have. Just go in and post it in the Q&A. We have some few minutes. So there's one question that's popped up in the chat. It was asking about, can you share which researcher or researchers worked on the non-tillable glyphosate versus tillage study?
So I presume you're referring to the Virginia State group, because the tillage versus non-tillage, that was, I did this one here. There was a group of people, and right off the top of my head, I cannot remember who the name of, but I'll just put in Angela Rourke is one that I'm fairly certain is one of the authors. So if you just Google this, pollinators, wildflower plantings, Angela O'Rourke, Virginia Tech, I think you're going to get to. I don't think she's at Virginia Tech anymore, but she was one of the people in that group.
Any more questions for Bodie? Since you're waiting, I have a question for Bodie. So how do you manage when there is a drought spells? Do you think about irrigation? How those things work on the wildflowers?
One of the things that I have tried over the years to see is how tough are these plants? In other words, non-irrigated conditions. So under mesic condition, how they would do. And I'm a firm believer of less inputs. So I treat them well when they're babies and when they're starting to get started. So give them a couple of times, 3 times, 1 inch of rain. And after that, just walk away. Of course, I don't put them in like June or July or August. I mean, I try to time it when you have lower evaporation, which is spring and fall. And that's how I treat that. So less is, you got to survive. If you're planting the small area, maybe 100 square feet, would you recommend 4-inch plants over seed? I would definitely go with plugs. Whatever the size of those plugs is not really as important. Just get some plugs, whether or not you grow those plugs yourself or you find them from a local place or you order them online. Make sure you match sun plant versus shade because again, that is important. There are plenty of plants that are for shade. And if you have shade, you have to go with those. If you have sun, make sure that those are sun type of plants. And I prefer perennials. I have also tried quite successfully. I've had some of the annuals also as transplants, like the Indian blanket and the partridge pea. And what happens, in my experience, is that those establish well and they start throwing seeds. So you actually get that area well combined between annuals and the perennials. Good question there.
Any more questions for Bodie? Okay, I'll call it all done. Thank you so much, Bodie, for this excellent presentation.